Welcome everyone. Today I'll be presenting work that we did with Cornelius Schröder and some colleagues in Tübingen, where we tried to understand the processing in retinal bipolar cells and downstream in retinal ganglion cells. So when a mouse sees an image, the light is projected onto the back of its eye in the retina, where it is processed by photoreceptors, then bipolar cells modulated by horizontal cells, and then passed on to ganglion cells and to the brain, and on the way modulated again by amacrine cells. In this work, we're using recordings of the bipolar cell outputs to this chirp stimulus, which is pretty standard in the retina literature. And here you're seeing two examples for two different types of bipolar cells. And we're also using recordings from the ganglion cells, again for two different types of ganglion cells. One disclaimer: we focus only on temporal processing using full field stimuli without any spatial structure, which means that direction selective cells, like the one down here, are not going to be well characterized. This is future work. Okay, so first, this is a model that we published last year, where we basically modulated or modeled the processing in the retina up until the output of the bipolar cell terminals. Including linear filtering, nonlinearity, a release stage, and different populations of amacrine cells that modulate the output. In this work, we're basically extending this model, and we're adding another, the simplest version, linear nonlinear stage that gives us, from that bipolar cell output, the output of the ganglion cells. We have three versions of this. First, a very simple one, which is really just linear, nonlinear, the release part of the bipolar cells, and then again linear, nonlinear to predict ganglion cell outputs. Then, naturally, the whole BCN output, including the amacrine cell modulation and the LN stage on top. And then the same thing, but with an added amacrine cell feedback stage that gives us more modeling power to model these effects. Okay, so comparing these models, maybe unsurprisingly, the most complex model is beating the other two. But I think it makes more sense to look at specific cell types, like here on the right, for instance, the on-mini alpha, between the BCN-LN and the BCN-ACLN with an additional processing stage for amacrine cell feedback. You don't see much of a difference. However, for the one below, the on-transient large ganglion cell, you see that you really need an additional stage of amacrine cell feedback to capture the response well. So this is suggesting that there are different types of ganglion cells that rely more or less on amacrine cell processing. Across the population, this is what it looks like. On the left, you have cells which don't care much about the additional amacrine cell step, whereas on the right, those which rely more upon it. Notice, however, that on the left here we have a bunch of the S cells, which again are probably not well captured by the stimulus, so take these with a grain of salt. We can also look at the connectivity weights between the bipolar cell types and the ganglion cell types and compare these to the literature. And what we find over here are some groups which are pretty well matched. And then on the right over here, some which are not super well matched. And again, these are the DS cells, so that might explain why probably the DS cells, you would find those weights rather if you're using a direction stimulus. We can also use these weights to predict where they stratify in the inner plexiform layer of the retina. And these are the same four cells as earlier, sorted according to how well they're matched. In the top, you see a good match, and in the bottom, you see a less good match. Okay, in summary, we present a complete network model of temporal processing in the retina, including bipolar cells, amacrine cells, and ganglion cells. We test the importance of amacrine cell processing between bipolar cells and ganglion cells. And we show the learned connectivity between cell types. And we can compare this to experimental data. In the future, we would like to run in silico experiments, make the amacrine cell pathway more involved and combine it with the bipolar cell pathway, and also, of course, use spatial stimuli to investigate the direction selective cells and others. And these are my collaborators. Thank you.